In part 2 of our series examining the structure of kidneys, we're going to take a look at microscopic structures found in the cortex and in the medulla of kidneys, and these structures, which are actually networks of microscopic tubules, are what we call nephrons. Now, the job of a nephron is to remove any solute, meaning any solid substance that is dissolved in the liquid part of blood, as well as a significant volume of water within our blood through a process that we call pressure filtration. So the way that pressure filtration works is inside of the cortex of the kidney, the high blood pressure coming from arteries forces the liquid part of the blood out, taking with it a large amount of solute that is dissolved within the liquid part of the blood, and then within the medulla, as well as in part of the cortex, the selective reabsorption, that being taking back in a large amount of water as well as the valuable solutes back into the blood, is what occurs. Now, if you are the human body, obviously you don't want to release all of the nutrients in blood, because then you would be urinating out amino acids and glucose and other valuable nutrients that we use for growth and for energy. And we also don't want to be getting rid of all of the water and all of the salt found within the blood, because maintaining the appropriate isotonicity, the appropriate balance between salt and water, is the other function of the kidney. For the remainder of this video, we're going to look at the structure of a single nephron in detail so that we can track the flow of nutrients released from the blood through pressure filtration and then reabsorbed into the blood via reabsorption. So if we start by looking at where blood enters the nephron, we can see here that this blood vessel, it would ultimately flow back to the renal artery, although this is more technically an arteriole because it's much smaller than the main artery that supplies blood to the kidneys. Now, renal arteries break off into smaller blood vessels, and these are what we call afferent arterioles. So afferent is a word that we've seen in the nervous system before, referring to a blood vessel that flows into something, and in this case the afferent arteriole flows into the primary location of pressure filtration, which is what we call the glomerulus. The glomerulus is basically a big, tightly wound ball of capillaries, which is located inside of a structure that we call the Bowman's capsule, and the Bowman's capsule is the opening, or the beginning rather, to the nephron. So we can see here this structure that looks kind of like a C that surrounds the glomerulus. This is the Bowman's capsule. And as soon as blood flows into the glomerulus, basically all of the solute that is dissolved within the blood, as well as some of the water, is squeezed from the blood vessels in the glomerulus into the Bowman's capsule like this. And this is the process of what we call pressure filtration, which we will look at in detail when we look at how the nephron actually functions. Now, from there, the blood that flows out of the glomerulus is going to have significantly less water and almost no dissolved content, whether it be nutrients or ions, flowing out of it like this. So this arteriole that leads out of the glomerulus is what we call an efferent arteriole, and as we looked at in the nervous system, whenever we see the word efferent, it refers to flowing away from something, and we can see that the efferent arteriole is flowing away from the glomerulus and breaks off into a complex capillary network that surrounds all of the nephron, and this is where the reabsorption of important solutes, as well as all of the water that we squeezed out, is also going to occur. Now, if we follow the filtrate, what was squeezed out of the glomerulus in pressure filtration, if this flows out of the Bowman's capsule, it flows into this crisscrossing network of tubules, which is what we call the proximal convoluted tubule, which again sounds a little bit overly complicated compared to what it actually does. Convoluted tubule. So in terms of each of these names, proximal simply means close to, as in close proximity, because it is the convoluted tubule that is closer to the Bowman's capsule and the glomerulus, and it's convoluted because the tubes don't move in a straight line for the purpose of maximizing surface area, but we can henceforth know this as the PCT. Now, in the PCT, the proximal convoluted tubule, this is where the overwhelming majority of nutrients are transferred from the nephron out of the proximal convoluted tubule back into the capillaries that surround it. Now, from there, the filtrate, which now should have most of the valuable nutrients, such as amino acids and sugars, reabsorbed into the blood, what is left of the filtrate is going to drain downward from the proximal convoluted tubule in a structure that we call the loop of Henle, presumably named after the person who discovered and characterized this. Now, the loop of Henle has a similar function to the proximal convoluted tubule, and that is the reabsorption of other substances. However, in this case, we're not concerned with the reabsorption of nutrients. We are concerned with the reabsorption of salt and water, which are done at different times. Now, because of its length and because of the difference in function between the two halves, we can refer to the half where the filtrate flows downward as the descending loop of Henle, and then the other half where the filtrate is flowing upwards here is what we call the ascending loop of Henle, and the distinction in function between these two halves will be revealed in the next video. Before we make it to the end of the nephron, we have yet another convoluted tubule, meaning another set of tubes that do not move in a straight line, again for the purpose of maximizing surface area. And because this side is farther away from the Bowman's capsule and glomerulus, this is what we call the distal convoluted tubule, although we can simply call it the DCT for short, and this is the final location in which any reabsorption occurs. And in this case, the reabsorption that occurs is going to be ions, for example, calcium, that is also important in bone structure and in action potential regulation. The regulation of calcium reabsorption, which is mediated by calcitonin and parathyroid hormone, is primarily controlled here, as well as the regulation of pH through the reabsorption and neutralization of H3O+ and OH- also occurs here to balance out any anomalies in acidic or basic pH that may occur here. Now, at this point, all of the reabsorption that will have occurred has occurred by the time the filtrate reaches the distal convoluted tubule, which means what is left is eventually going to become urine, because it flows into this structure here that eventually leads to the ureter, and this structure here is what we call the collecting duct. We call it the collecting duct because we can see that it isn't just this nephron that flows into it. We can also see a number of other points where multiple nephrons within the kidneys join and feed their filtrate into this collecting duct here. And the final job of the collecting duct is to balance out how much water and how much salt is within the urine to make sure that we are getting that isotonic concentration before it drains out of the nephron and eventually into the ureter, where eventually it is going to make it into the bladder and stored until urination takes place. In the next series of videos, we're going to be taking a much more detailed look at the nephron and looking at how pressure filtration and specific reabsorption of specific solutes and water occurs at specific locations within the nephron itself.